All right, where he is, the man of the hour again on Healthcare Unfiltered. The good news, it means I did not mess things up the first time around. He was with me last year, so he agreed generously to come back on Healthcare Unfiltered, the star of the show who pretty much every week has a paper coming out somewhere. Either way, Vivek, I've lost track. I mean, I don't know what, we don't have time for all that stuff. It's a little bit too much. Welcome to Healthcare Unfiltered, Vivek. I'm a big fan, and we're going to talk about a lot of nice things pertaining to biomarkers, precision oncology. But I want to really focus a little bit more on the regulatory and synthetic control arms that you were talking about on social media. Well, let's start by the usual stuff. A little bit of introduction about you and so on, where you came from, and how did you end up at MD Anderson?
Oh, thank you so much, Shadi, for having me back in this podcast. It's always, again, I started listening to this podcast when I'm driving. I think this is, although I go to the YouTube videos, I started, listening to these podcasts. So, some of them are phenomenal. I think especially, again, we tend to work in academia and we tend to work in silos, think in silos. I think the Healthcare Unfiltered has opened up silos. Social media one way is opening up silos, and podcasts, give us more, in terms of their real, thought process. So I think thank you for, doing this.
No, I appreciate you. Very kind.
Doing this. And then people need to know the right information as well. Right? People who are listening to, especially people who are not involved in clinical trials, involved in drug development, or the lay audience, they need to know the right, information from different points of view.
I appreciate your support.
So just to give a brief introduction of me, Vivek Subbiah. I'm currently a medical oncologist in the University of Texas MD Anderson Cancer Center. And I did my training, medical school in India. I moved here and did my residency in Cleveland, Ohio. I did a combined medicine and pediatric residency there. And I never wanted to be an oncologist, but things so happened that I loved doing clinical research, and I wanted to do clinical trials. That's why I started a career in a place that trains one for clinical trials. I did a combined hem-onc, pediatric and adult hem-onc fellowship at MD Anderson. And 2012, I started as a faculty in MD Anderson in the early phase drug development program. And I'm currently the Center Medical Director of the Early Phase Trials Program. And just before the pandemic, I took on another role as Executive Director of Medical Oncology Research in the MD Anderson Cancer Network. So in my brief, time here at MD Anderson, it's been, rewarding to, lead several practice-changing clinical trials, mainly precision oncology clinical trials to FDA approval. Right from rare diseases like anaplastic thyroid cancer, BRAF, recently the 2 selective RET inhibitors, and to recently the tissue agnostic BRAF inhibitor drug development, FGFR, lurbinectedin, and among several others.
Well, it's been wonderful, truly, watching your productivity, and to me, I honestly don't judge productivity by the number of papers, because frankly, you could write 200 review papers with a nice CV. I judge productivity into the impact of what you are actually doing on clinical care and advancing research.
Absolutely.
And there's no question you have had amazing impact, and I am glad that you went into oncology. I think patients are served very well by doing that. So there's so much we could talk about. When I had you on the show, I think it's been close to a year. I was looking when you were on, it was really last spring of 2021. So it's been a while.
It seems like a decade because of the pandemic. The time is in a warp speed.
It's crazy.
Yeah.
But we talked about precision oncology in general. I think we were just very in general. What I want to do differently today, I want to talk a little bit about precision oncology, but then I want to really segue into when you design clinical trials like the ones that you were involved in that led to the approval. I wanted to take listeners through the process that happens. You're an investigator, there's a drug maker and a sponsor, there's the FDA, the regulatory process. I mean, how does this actually go into until you go to the in front of the FDA and the FDA grants this, the type of studies, because I mean, the trials that you're doing are always small cohorts. They're not really, and I think that's really where the field is going. So let's start by explaining in your own words, how do you define precision oncology today?
Again, precision oncology is delivering the right treatment to the right patient at the right time. So let's go back, say 2 decades ago. In the 1990s, they said, the human genome can never be sequenced. And early 2002, it took almost $300 billion and a decade to sequence the first human genome. We are learning. So right now, today, as you know, and others know, we can sequence, do a whole exome, a whole genome for $5,000 in the US. And if you go to China or India, you can get it sequenced for less than $100. So again, clinical next-gen sequencing has been rapidly evolving in the last 2 decades. And at the same time, there has been an explosion in terms of drug development, right? The immunotherapies and targeted therapy. So the clinical next-gen sequencing helps us identify targets across multiple cancers. And, then at the same time, in the last two decades, there have been a revolution in the drug development paradigm, right? We have immunotherapy. We have a lot of targeted therapy. Let's take, for instance, the case of lung cancer, which has become the poster child for precision oncology. So a decade ago, when patients were diagnosed with lung cancer, it's either non-small cell lung cancer or small cell lung cancer. Non-small cell lung cancer get the same chemotherapy and we get the same outcomes. With comprehensive genomic profiling, we've identified today, at least we have 10 genomically defined subsets of lung cancer that can be benefited from these precision medicines. So again, we are here and lung cancer has become 1%, disease. Like for instance, the RET inhibitor studies that I've led, it's 1 to 2% of non-small cell lung cancer. And we have effective drugs to go after these diseases. So now, a decade later, we are sitting with the success of these therapies. At the same time, there is a lot more work to do for 80 to 90% of patients who do not benefit from these precision therapies.
So, but do you envision that histology and origin of tumor are going to matter in 5 years? I mean, are we getting, because we're getting so many of these new drugs that are being approved tissue agnostic based on the actual driver mutation or whatever the biomarker is. Is that where we're heading? I mean, is that what's going to happen?
No, I don't think so. Because if you think about the US FDA approval process, the US FDA has approved drugs to treat patients with tumor types based on single anatomic sites, such as kidney cancer, melanoma, rather than biomarker alone. This standard approach is based on a number of factors, including heterogeneity of drug effects in different biomarker positive subtypes. Additionally, drug development for some drugs were primarily directed towards a specific, genomic type in a specific tumor type. So the thing is that most genomic alterations are shared across multiple cancer types. So there will be, genomically, diverse, targets like tissue agnostic target drug development, but there will definitely continue to be 90% of drug development is going to be histology specific.
Got it. So there will be histology, the majority will be histology specific in your opinion, even as we move forward.
Absolutely. Majority will be histology specific because that's how clinical trials are designed. That's how, institutions are set up. That's how, trials are designed. So histology agnostic drug development is interesting, right? So the first ever drug to, receive a histology agnostic drug approval was pembrolizumab, an immunotherapy for, microsatellite unstable cancers across tumor types. And interestingly, this was a first drug approval. And following that, we had NTRK fusions that were seen across tumor types. And larotrectinib was the first FDA-approved targeted therapy towards as a histology agnostic manner. And entrectinib followed suit. And then pembrolizumab again was approved for high tumor mutation burden high. And recently we had the BRAF drug approval. So again, these drug approvals in a histology agnostic way seem to be minority. And histology specific drug development will still continue as a major clinical trial paradigm.
All right, so that's helpful. I actually expected the opposite answer, so that's why I asked the question. And I'm glad that it seems like we're going to still rely on the bread and butter oncology and so on. I apologize for the noise. Probably there's a little bit of noise coming through the system for the listeners. Let's go through the regulatory process. Let's try to understand what you have worked on over the past several years. Is you brought in so many drugs through the FDA process, through the FDA and regulatory process. Take us through one example. How does it start literally from A to Z? Somebody calls you or you call the company and say it's investigator initiated. How does it go and how do you agree on finding the actual study? Do you discuss it with the FDA? Just take us through, regulatory process for your type of studies for dummies.
Oh, absolutely. Thank you so much. So again, everyone is smart. I wouldn't call dummies. I think when we think
for me, it's just me.
We are dummies because I feel I'm a dummy because I'm not able to get every drug approved or every clinical trial successful that I do. I'm still learning. Again, for the lay listeners, there are multiple ways clinical trials can be done, right? Clinical trials itself are divided into phase 1 studies, phase 2 studies, and phase 3 studies, right? And if you see, there is so much attrition when we move from a drug from phase 1 to phase 3. So what do they do, right? So all academic institutions or scientific centers and labs test, do something called drug screening. They test these new drugs in petri dishes in something called what we call as cancer cell lines. So again, tens and thousands of drugs are being tested like that every day in so many labs. And among those, there are a few drugs that show some activity in these petri dishes with cancer cell lines. They think it is promising, and then they take it to, mouse models, like preclinical studies. That's what we call as preclinical studies. So the preclinical studies, most of the drugs, if there's 10,000 molecules make it to preclinical studies, most of them will be dead by then. So 100 molecules make it beyond the mouse model studies, then they move into something called large animal studies, which we use dog and or monkey models. So when they move to large animal studies and monkey models, if the drug is toxic, then the drug is shelved. And if they pass through all these phases, they make it to something what we call as first in human studies, right? Phase 1 studies are first in human studies. So when first in human studies, what is the purpose of first in human study? To check the safety and toxicity of these drugs. We don't know the human dose of these drugs. So what essentially, we do is, let's take, for instance, a non-oncology drug, say Tylenol, right? For a patient to be alleviated from headache, if you need 500 milligrams, how did we get to the 500 milligrams of the drug? We did not know that. So they would have tested that in monkeys, and they would have known that the drug is not toxic below a certain level of the drug. And then based on certain calculations, they come up with the first in human dose. And glad that people in the 50s and 60s have done all these calculations for us. So we are able to guesstimate and calculate the first in human dose. So what they would start as, say, they would start at 10 milligrams. So the conventional trial phase 1 design is a 3+3 design. They will start at 10 milligrams and they will enroll 3 patients in that. They will wait for a month. If the patients do well on the 10 milligrams, then they go up a dose level. They go to 20 milligrams. So this process, iterative process, goes on until, the target, until the patient develops toxicity. So they will measure labs, they will do scans and everything on these patients. And if there are any abnormalities, say liver function abnormalities, what we call it as DLT, or dose limiting toxicity. So they go, say, from 10 milligrams, they went all the way up to 600 milligrams. And at 600 milligrams, they saw liver abnormalities. That is a DLT. Then what they do is they go back down and dose more patients in a lower dose level, say 500. So then if that dose is safe, that becomes something what we call as MTD, maximum tolerated dose, or recommended phase 2 dose in this case. So then that is the phase 1 study. The phase 1 study ends there, and because we know the dose, and then the next process is a phase 2 study. What's a phase 2 study? So we know the dose of the drug. Now we are checking the efficacy of this drug and also the toxicity of this drug, because we are going to be treating more patients at a specific dose level. And beyond phase 2 studies, if the drug shows efficacy data, so for instance, in oncology, cancer drug, if the drug shows that it is active against a particular disease, then phase 3 trials are done using what we call as randomized control study designs, where this drug that we are testing is randomized against placebo or standard of care. Again, this is a traditional pathway of drug development. If the experimental drug shows benefit versus standard of care for that particular disease, or versus placebo, if the drug shows benefit, clinical benefit, they do all these statistical designs, and then they go to the FDA and show the data. The FDA looks at the data, the efficacy, toxicity data, and then based on that data, if the drug is beneficial, the drug received what is called as FDA approval. That means that the drug company has marketing authorization for the drug. So that is the traditional model or drug development paradigm in oncology: phase 1 study, phase 2 study, and phase 3 studies. So this process.
Vivek, before you go into the other process, how long does that traditional thing takes, just to give listeners an idea in terms of the duration?
So the traditional process in oncology takes anywhere from 10 to 15 years. You see that, right? Phase 1, doing preclinical studies, phase 1 studies, phase 2 studies, and phase 3. The traditional model, takes 10 to 15 years in oncology. So this is what was used to being the conventional, phase 1, 2, 3 trial. So, this is the basics of, clinical trial drug development in oncology. So there are several ways of studies where you asked how we can do clinical trials, right? So one is called the investigator-initiated clinical trial. So investigator-initiated means an academic centered PI has an idea that this drug may work in a particular cancer, either alone or in combination with any modality, or a combination with drugs. So then I need to show some preclinical data, right? I work with my collaborators or do it in my own lab that this drug is supposedly active in this disease. And then I have to apply to pharmaceutical company because they have the drugs. So, and they give access to the drugs and they provide funding, some funding to do the investigator-initiated trials. So, that is a pharma-funded investigator-initiated trial. And, it's usually done in a single center or 2 or 3 centers. There is another way of doing investigator-initiated trials is through NIH. The NIH and NCI has this program called CTEC. So wherein all the pharma companies that have drugs, give access to all the drugs in NIH. And if the concept and the scientific rationale of a study is compelling, has good preclinical and or clinical evidence, we can pitch in something called the letter of intent to NIH. If that is approved, then we can do a clinical protocol with NIH. The good thing about doing a trial with NIH is you can get access to the drug, plus you can combine 2 drugs from 2 different companies. So it's always hard as an individual investigator when you want to combine 2 drugs from 2 different companies, because most of the legal language from academic institutions and many other companies, is quite challenging. So it's easy through the NIH to do that because both the companies have given their drugs to NIH, and if it makes sense, you can access both the drugs through this mechanism. So those are, some of the few mechanisms of investigator-initiated studies. And another, mechanism that we can do studies are called through cooperative groups. Cooperative groups like SWOG, ECOG, they do, studies as well. Those are big randomized studies across major academic centers. So in the last 20 to 30 years, what has really come up is what we call as sponsored studies. Sponsored studies are, pharmaceutical companies, right? Because they make the drugs, they own the drugs, and it is of their interest to develop these drugs so that they can get FDA approval. And they want to make sure that this process is seamless and fast. They do these clinical trials. So sponsored trials are done by pharma companies, right? So in sponsored studies, what happens is they reach out to academic investigators in multiple centers, and they form something called the steering committee. And the steering committee provides the input for the study design to pharma companies. And based on that, the study is designed. And then, if it's a phase 2 study, it's done across multiple centers, and the pharmaceutical company engages several academic centers. And as and when the trial progresses, the data is analyzed. If the data shows that the data is promising, then they analyze the data, they present it in national meetings, they put the data together. And then what happens is, once they move on to phase 2 or phase 3 studies, they go to FDA with the data, and they ask FDA to approve the drug so that they can start authorization. There are multiple steps involved, and I think FDA approval process can itself be an entire topic for another day.
And is it what you're seeing right now is a lot of these tissue agnostic biomarker driven studies are more coming as pharma sponsored trials? They have a target, they have a drug, and they come to investigators and say, we want to get this drug through the regulatory approval, and they design that trial. Is that what's going on? I want to try to take one of your trials. Let's say the last one you just did that just led to the approval of the BRAF plus the MEC, yes. I believe, right? Let's yes. go through that. How did this come about, and how long did it take until we got the FDA approval?
Yeah, BRAF is an interesting target because, as you know, BRAF is prevalent in melanoma. 50 to 60% of melanoma, patients with melanoma harbor the BRAF V600 alteration. And we have 3 combination drug approvals in this space. And BRAF as a mutation, BRAF V600 mutation is seen across multiple tumor types. But in the beginning, not many pharma companies wanted to do a tissue agnostic study in BRAF because, one, the thought process and trial process was not there a decade ago. And most of the academic centers were histology specific. And if they want to, have these studies across diseases, they did not have a specific center to house these studies. And another important aspect of not doing a histology agnostic study in BRAF a decade ago was the data from colorectal cancer. Although in melanoma, there was striking promising responses with all these BRAF inhibitors, in melanoma, the response rate was negligible. So at the same time, they went back to the lab and found out that EGFR is an innate mechanism of resistance operating in colorectal cancer. And the addition of an EGFR inhibitor, in this case, cetuximab or panitumumab, overcame that resistance pathway. So that operating mechanism was only present in colorectal cancer. Because of that, I think companies did not pursue tissue agnostic approval in BRAF V600 cancers. When we designed the study, say in 2012, this was called the ROAR, Rare Oncology Agnostic Research. We picked up 9 solid tumors with no FDA approval and unmet need. And we enrolled patients in these 9 different baskets. As long as these tumors, rare tumors, harbored the BRAF V600 alteration, they would receive 2 oral drugs, dabrafenib and trametinib. The primary objective was, objective response rate per investigator, and it was later confirmed by, central review. And we found, again, when we started doing the study, we saw that there was this cancer called anaplastic thyroid cancer. Anaplastic thyroid cancer is one of the most lethal cancers known to us. It's a rare form of thyroid cancer, and the median survival for this anaplastic thyroid cancer is less than 6 months. And we knew at the time that some of these patients with anaplastic thyroid cancer harbor the BRAF V600 alteration. And I was probably one of the first few physicians in the world to treat a patient with BRAF inhibitors in anaplastic thyroid cancer, and we saw promising responses. And because of the promising responses in that particular cohort, in that Rohr basket study, and there was no FDA-approved drug in anaplastic thyroid cancer, based on a data set of, say, 23 patients, we were able to get an FDA indication in this rare disease based on the rare oncology agnostic research. Again, that was a win. And then we continued to enroll patients. And recently we enrolled around 36 patients globally. And we confirmed the definitive benefit of this combination in anaplastic thyroid cancer. So beyond anaplastic thyroid cancer, we had these 9 different cohorts that were also accruing. We had patients with cholangiocarcinoma. We had patients with glioblastoma, brain tumor, again, another challenging tumor. We also know that glioblastoma, low-grade and high-grade gliomas, harbor this BRAF alteration. So we enrolled patients in that cohort. We also had a hairy cell leukemia cohort. We had other cohorts as well. So as time passed on, we found out that colorectal cancer is an exception. And there are so many different cancers that do respond to BRAF V600 inhibition. In parallel, the NCI match study was established and patients were accruing across the nation. And NCI match, interestingly, also had an arm with BRAF V600E altered patients with the same drugs, dabrafenib and trametinib. And we saw that NCI match confirmed our raw data that this dabrafenib and trametinib combination showed activity across multiple cancers. So we aggregated both this data together. At the same time, we also aggregated data from pediatric clinical trials. Again, as a tissue agnostic, we wanted to be age agnostic as well, because BRAF is also seen in pediatric cancers. So based on the aggregate and totality of data showing that colorectal cancer is still an exception with BRAF V600 alteration, we were able to apply for an FDA approval, for across all solid tumors. Again, we need to note that this is not the first approval for this target, right? So the drug has already been approved in melanoma, already been approved in non-small cell lung cancer, as long as they have a BRAF V600 alteration, and anaplastic thyroid cancer, and now across all solid tumors. Again, this provides a way in which drugs can be authorized, drugs can be approved by the FDA. Again, who will benefit from this tissue agnostic approval? Rare cancer patients, right? So patients with, say, for instance, cholangiocarcinoma, or salivary gland cancer, or pleomorphic xanthoastrocytoma that we have in those studies. How will a patient sitting, say, in Dakota or Kentucky or Alabama, who don't have access to a major cancer center, access these drugs if they have a BRAF alteration? So they need to ask for insurance approval, which may seldom be approved because it's not approved for the indication. And number 2 is they need to file for a compassionate access. Its paperwork takes a long time. So again, this FDA approval provides access to these rare cancer patients.
So Vivek, you're talking the accelerated approval pathway.
Yes, yes.
So that is a nice segue into the accelerated approval pathway, because basically the company starts the process with the idea that they are going to go through the accelerated approval pathway.
Yes.
Correct?
Yes.
So they have the biomarker.
Based on the early signals of activity, right? So, we need to know that the drug works.
But in designing the trial that they are going to use for the accelerated approval pathway, do they have a discussion with the FDA before, and they say, okay, this is what we are designing.
Yeah.
This is our phase 2 study. We are going to enroll 100 patients, various tumors, have the BRAF V600 mutation. And our goal, if we see a signal, we are planning on applying for, we're planning to get an approval. Is the FDA engaged in that dialogue at this point or not yet?
In some cases, yes. I think right now, we are more and more, we know these targets now shared across multiple cancers. I think the companies, when they design, especially these phase 2 basket studies, they do sit and discuss with the FDA and ask, even seek guidance and counsel on what should be the response rate cutoff if, for a potential indication. In many cases, what happens is the trials are designed, the trials are done. Midway in the trial, we see interesting signals of activity. In that case, we go to FDA with a meeting called a Type C meeting, an ad hoc meeting, showing the data to explore possibility to see if there is a pathway for a drug, accelerated approval for this drug. I think both ways, it can be done both the ways, but I think more and more they are encouraging that they want to have the discussion with FDA before designing the clinical trial, when you're designing the clinical trial, rather than later.
So, but as part of the accelerated approval pathway, and correct me if I'm wrong, as part of that approval, you must conduct post-marketing study.
Exactly.
And that is technically should be a randomized control study, because you're saying, okay, I believe this drug works, and you need to show that in a randomized controlled fashion. And the idea is that if it doesn't work in the post-marketing setting, then probably what you saw was more noise and not signal.
Again, yes and no, because there are some exceptions, right? For instance, in the case of extremely rare diseases, it may be infeasible to conduct randomized studies, and it may not be, we may not have even clinical equipoise to randomize patients to standard of care, right? There have been several instances of FDA approval. Again, those are exceptions, rare diseases, but most of the programs with accelerated approval have a post-marketing commitment of a confirmatory study, which can be a randomized study, or which should be a randomized study.
Okay, but it's not always a randomized study.
It's not always a randomized study.
So what type, I guess, take us through the type of studies that could be conducted in the post-marketing setting for accelerated approval, and they remain acceptable by the FDA, because I believe that is where you start talking about synthetic control arms.
Exactly.
So yeah, take us through the type of studies where I got my drug through the accelerated approval pathway. I got the folks are able to prescribe it, but now I must do this post-marketing trial. What type of post-marketing studies are acceptable to the FDA?
Absolutely. I think, this is, we are all in the learning experience phase on this, right? I think I wouldn't say what are acceptable to the FDA, but again, these are some of the ways that, supportive evidence for the drug can be, handled, after accelerated approval. So although randomized control trials allow for comparison of treatment arms with minimal concern for confounding bias for known and unknown factors, randomized study, as I said, is not feasible in certain disease settings, right? When randomized design studies are not possible, what we call is ECA, external controlled arms, or also popularly known as synthetic control arms, incorporating into the study design can be an effective way to expand the interpretability of the results of an experimental single-arm trial by introducing the ability to carry out a formal or informal comparative analysis. Again, as and when, we are learning, right? Like, these are emerging healthcare technologies. And, they've been slow in the last decade to gain traction because there is still confusion and skepticism about external control arms. Why do we have the skepticism about external control arms? Because it stems from lack of precedence, right? And there is also not step-by-step guidance and data to quantify the impact on drug development and clinical outcomes. So this uncertainty has, given us several myths around the synthetic control arms, because it's always a fear of the unknown. And, as I said, external control arm, we call it as ECA or SCA, synthetic control arm, are based on what we call as RWD. Again, this taxonomy is again and again repeated in the real world research. That's RWD. What is that? It's real world data. What is real world data? Real world data is data that is pulled from the electronic medical records and medical claims, medical bills. And, in non-oncology fields, they are using even wearable technology and wearable devices to access real world data. And there are several other sources of real world data. And what these synthetic control arms or external control arms are being used to. So they are used to now primarily, again, they can't support the approval. They can provide, some supplementary evidence to a single arm study. And also they can also help in terms of label expansion of drugs, especially in rare diseases. And even for the pharma company for go, no-go treatment decision for studies, right? When they want to compare that with real world data, if their drug doesn't fare well, it can be used for, a go, no-go decision and, and not investing on the drugs. So again,
so I think Vivek,
yeah.
So I think the label expansion is probably self-explanatory.
Yes.
Right. You see the drug being used in the real world.
Yes.
You may see an activity in a tumor you didn't think about. And then you take that data to the FDA and say, hey, look, I did not think the drug works in sarcoma, but it's being used off-label and I have a data on like 500 patients and it's working. And they may give you a secondary indication. Or they may give you an expanded label that, for example, the label is for, let's say, theoretically, for people younger than the age of 75. And then you show in the real world data that they work in patients over 75 and they expand the label. But that neither of this, the expanded label or the secondary indication, these are not comparison, randomized control. I guess my question is, how do you use RWD or the synthetic control arm in a randomized way in the post-marketing era? Is that what is used or no? Like, do you actually take a cohort from the real world and say, and you say, how do you do it? How do you integrate this as part of a post-marketing randomized setting?
Absolutely. I think that's a great question. So this is now well documented, right? Now, 2 years ago, if you asked me this question, this is not well documented. But now this is well documented. So external control arms are well documented in the post-approval setting. In the US healthcare system, drug makers, seek reimbursement from payers by conducting database studies, post-marketing studies in which external control arms are often leveraged for price negotiations and coverage. And Europe, after they go, FDA, it's only one step, right? It's FDA approval and then drug the drug is reimbursed. In Europe, they have something called the EMA approval. And then there is a lengthy process in each country that they need to go through this process called HTA, Health Technology Assessment. So in Europe, the manufacturers, the pharma companies need to submit lengthy dossiers and memos to each country's health technology agency to seek insurance coverage and ensure patient access. So in these situations, the external control arms provide the foundation of comparison between active treatment and a control cohort. Again, these are still in the early stages of adoption. I think I can share with you, some of the early external control arm or synthetic control arm data that led to even FDA considering some data for FDA approval in rare diseases. And also recently, we published how we address challenges with real-world synthetic control arm to demonstrate the comparative effectiveness of a selective RET inhibitor in non-small cell lung cancer. So, as you know, recently, in the peak of the pandemic, we conducted a clinical trial called ARROW, which is an open-label multi-cohort phase 1, phase 2 study that demonstrated pralsetinib, a highly potent selective RET inhibitor, was efficacious in RET fusion positive non-small cell lung cancer patients. So given the promising results, in 2020, the drug pralsetinib was FDA approved for treatment of RET fusion positive non-small cell lung cancer patients. So as you know, RET fusion lung cancer patient is just 2% of non-small cell lung cancer, 1 to 2%. So there is a frontline randomized study ongoing, but it's going to take a long time for us to build evidence from a frontline randomized study of pralsetinib and first-line standard therapy. In the background, one of COVID infection and also an unprecedented pandemic, we have a lot of recruitment challenges. To fill this evidence gap, one of the goals of this study was to investigate the relative effectiveness of pralsetinib by comparing outcomes for RET fusion positive patients received first-line pralsetinib in the ARROW study. So we have the data. That data has already been published in Lancet Oncology, right? So we use that first-line pralsetinib data with synthetic control arm derived from the real-world data, right? We drew upon real-world data to construct synthetic control arms for comparison, where running a randomized trial in this case was infeasible, or randomized study is ongoing, but it might take time for us to get the results. So in order to satisfy the requirements of HTA, or in order to even be us being comfortable when we offer this drug to patients, right? When lung cancer patients come, we know if the patient's tumor harbors the RET alteration. What do you want to give? Standard chemotherapy with pembrolizumab in combination with standard therapy, or pembrolizumab, or this RET inhibitor. So we wanted to build confidence in us treating this. So we did this, sort of a randomized study with synthetic control arm. And, we showed our goal was to investigate the comparative effectiveness of the RET inhibitors related to other therapies in the first-line non-small cell lung cancer. So one of the main challenges in real-world data and building real-world evidence is the bias, right? There's so much bias that we need to be cognizant of when we design these synthetic control arm studies. So that is why we want to do a randomized study. What are the biases that we see? We see confounding bias, selection bias, performing bias, detection bias, attrition bias, and time trend bias. So all these, again, we can mitigate and we can address some of these concerns by some statistical analysis, mitigating, how we can, that's what we've showed in our Nature Communications paper, is how we are able to address some of these biases when we randomize, a clinical trial single arm study with a real world, a synthetic control arm.
I'd like to maybe address a few of these issues with synthetic control arms, because you mentioned some of the biases. I think one of the biggest biases that you will hear amongst clinicians, investigators, is that in the real world there is no uniformity into how you define response, or even how often you do imaging studies.
Absolutely.
I mean, once you go in the real world, you may decide to scan your patient every 2 months. I may do it every 3 months. I may literally adhere to the RECIST criteria, and you and I know in the real world nobody is really measuring with a ruler RECIST, and they're really probably saying if the node is shrinking and the patient is doing okay, then they're responding and I'm moving on. So how do you address these two factors? Assessing the response, because you're really not using a uniform way, and then addressing what, let's call it progression-free survival, because really frankly, the PFS is contingent on how often you are assessing progression with imaging. So if you're really not doing it enough, you're probably going to have artificially prolonged PFS. So how do you address these two biases?
Absolutely. I think that's a great question. So in that study, particularly, I think if you go and read that study, we directly compared the overall survival, the PFS, and also importantly, time to treatment discontinuation, right? So again, oncologists are not going to continue a drug if the patient is not benefiting from the drug, right? Either clinically or radiologically. So again, we also used another endpoint called time to treatment discontinuation, and we directly compared for a selective RET inhibitor versus monotherapy pembrolizumab versus chemotherapy plus a combination with pembrolizumab, and we built in something called tipping point based bias analysis, assuming non-random, missingness for, ECOG performance status and impact of metastasis. And again, what we always know is that real-world patients have often a poor performance status and maybe even borderline labs than patients who enroll on clinical trials, right? So again, we took all these, measures of confounding into our analysis. When we did the sensitive analysis, we went back and forth with brilliant, statisticians who delve into the, real world, database and on a single arm cohort clinical trial. Again, being a rare mutation, 1 to 2% of lung cancer, along with limited uptake over time, we expected that there would be less patients in the real world. So that's why we matched this real world cohort with patients without RET fusions as well, and we showed that even if we adjust for all these biases, all these mutations, a selective RET inhibitor in the frontline setting was superior, or in fact you can say non-inferior to pembrolizumab or pembrolizumab with chemotherapy, so that we have better confidence in treating these patients as clinicians. And also, importantly, health technology agencies can say, yes, they have some data, supportive data, to show that in the real world setting, these patients are benefiting from this treatment.
So maybe a couple other questions, and I can't believe we're almost at the top at the hour. This is crazy. I can't get enough of talking to you. But once you have this accelerated approval, and you are now in the process of designing the post-marketing study, whether it is a randomized prospective RCT, whether it is comparison to a synthetic control arm from real-world data, is there a dialogue with the FDA by you go to the FDA and say, this is what we are thinking. If we are proven correct with this design, then we expect a full regulatory approval?
Absolutely, absolutely. I think this is in fact when the drugs receive approval, it is a mandate by the FDA to drug companies that they have to follow certain clinical trials. The clinical trial design is discussed. Many times the clinical trial for the randomized studies or post-marketing studies are designed even before the drug is receives approval, and that's when confusion ensures. Again, time and again, we've seen that it is always good to talk to the FDA and regulatory authorities to make sure that this trial, if successful, would be the confirmatory study.
So what happens if the confirmatory study post-marketing does not pan out? What happens then?
That's a great question. So I think, in many cases, there are two things, right? One for common diseases, one for rare diseases. So in common diseases, it is the company has to withdraw the marketing approval, and the company has to withdraw approval. And they have to perform due diligence and make sure that they have to withdraw marketing approval. Many times what happens is the trial that is done may not be the confirmatory trial that would be the perfect trial. So then in that case, even if the trial is negative, it means that the drug it's not a failed drug, it's a failed trial. So in that case, they can go back to FDA and ask them for some more time to design the proper randomized study so that this issue can be answered. In some cases, especially in rare diseases, sometimes the FDA may mandate confirmatory studies, but it may not be possible or feasible for these studies to be done. In that case, the company needs to have an open dialogue with the FDA and also seek, other, mechanisms of, post-marketing continued approval, like use of external control arms or, external control studies. There have been several instances recently, regulatory case studies that even the FDA put out, that they've used the synthetic control arms for, taking the regulatory decision. There have been decisions like a drug like selumetinib, which is a MEK inhibitor. There is a rare form of, pediatric cancer called NF neurofibromatosis type 1 with inoperable plexiform neurofibroma. So they used previously conducted clinical trials as sources of external control data, mainly to establish a natural history of disease. And apparently they just wanted to make sure that there is no, spontaneous regression in these benign tumors. And they used that. And recently there was another drug approved for bladder cancer, edafitinib, for FGFR gene selected alterations. And FDA used patient level EHR data from US community-based clinics to establish the natural history of disease. And we see all these external control arms also being used in also looking into contribution of components. For instance, as you know, pembrolizumab, Keytruda plus lenvatinib was approved in endometrial cancer, which is not MSI high. And, people wanted to know the contribution of pembrolizumab and lenvatinib. So in that case, they used other trials with monotherapy pembrolizumab and monotherapy lenvatinib for contribution of the components, and what the regulators call it as isolation of treatment effect, that the combination is beneficial than the individual components alone. So there are several use case scenarios in terms of regulatory pathway for these synthetic control arms. And again, this is not exhaustive, right? So we are learning more. As with any novel technology, right? The real world data is completely imperfect today, but it is a definite hurdle that can be overcome. So as you said, physicians in the real world are not following clinical protocol. There'll be always less consistency in the real world related to clinical trial data. So to overcome potential bias, we have brilliant biostatisticians, and so many of us have developed robust methodology and statistical approaches. Again, we are also using inclusion-exclusion criteria like we use in clinical trials in the real-world data to avoid cherry picking and augmenting real-world data to closely mimicking the information that we see on clinical trials. So again, we are just still scratching the surface for synthetic control arms. It's still in very, very early stages of adoption. There are so many stakeholders, are not certain about their long-term potential for clinical research, but with definite buy-in, especially from regulators, especially from the payers, with creativity from academia, creativity from statisticians, creativity from industry, I think clinical trial sponsors will quickly start to see the benefits of these real-world synthetic control arms in clinical trials.
So have you, two questions, and I promise I'll let you go. When you get the accelerated approval, you still don't have the full approval, right? You have the accelerated approval.
Yep.
But as a prescribing physician, there are no limitations, right? I mean, you can do whatever you want. I mean, technically, it's just a regulatory issue. It's like a regulatory stamp. But when there is an accelerated approval, as a doctor, I can prescribe that drug whenever I want. So how long does it take from accelerated approval until full approval? And then I'll tell you why I asked that question.
It depends, right? Sometimes there are drugs just with accelerated approval and that haven't had a full approval even after like a decade. And there are some drugs that have been withdrawn from accelerated approval because the confirmatory trials did not pan out after like even 5 years. I don't have the specific numbers, but usually from accelerated approval, majority of the drugs, at least in precision oncology, not majority, almost all the drugs have been confirmed with full approval in the precision oncology setting. And I keep track of all the precision oncology drugs. Immunotherapy, as you see in that paper, is in the New England Journal paper, it's a wild, wild west in terms of immunotherapy. But precision oncology accelerated approvals, right, have been, I think accelerated approval has been built for precision oncology. Just like precision oncology is, directing to the right treatment to the right patient at the right time, accelerated approval has been the right pathway for precision oncology at the right time.
I mean, I think that is another podcast. I do think that one of the issues to accelerated approval that people contend is that not every drug that does not fulfill its promise in the post-marketing era gets withdrawn. And you said that yourself. And then the question would be, well, if you went accelerated approval and you did not show this later on and you did not withdraw, then are we like, are we scamming the system almost?
No, that's a good question. I think, that's unique in oncology, right? Because, sometimes we are stuck with a drug based on single arm data that shows a response rate and some overall survival data. And the confirmatory trial is not the confirmatory trial one would think of. It's a random, random study that was, touted to be the confirmatory study in combination with some other agent. And that confirmatory trial in combination was negative. That means the trial is negative, the drug doesn't fail. So I think, if in the case of a disease that doesn't have an FDA approval, withdrawing that drug from approval will in fact, patients will lose access to the drug, right? In the real world, I think, why does FDA need to approve a drug, right?
But that's okay though, Vivek. I mean, if you think about it, if you losing access to a drug that we may not know if it helps people is okay.
In rare diseases, again, again, there are always exceptions to the rule. Again, we were talking about this morning about a drug called clofazimine, which is used in pediatrics in a relapse refractory setting. And apparently it's been...
I wrote a paper on clofazimine in 2005. You were probably in high school. I had an investigator-initiated study on clofazimine in refractory and relapse lymphoma. It's in cancer.
Is that right? That's awesome. I will go ahead and read it. Yeah, no, absolutely. So that drug, apparently it's approved in pediatrics via an accelerated approval mechanism. And never was a confirmatory trial was done. I don't know the reasons. I need to look up at the reasons. And if that accelerated approval is pulled, again, people use clofazimine in the relapse refractory setting if there are no drugs, right? If someone's going to pull that approval, pediatric patients with this rare, leukemia in the relapse refractory setting may not have access to the drug. Of course, having a drug approved means a physician has access to the drug. So no physician is going to give clofazimine in the frontline leukemia, especially in pediatrics. So again, in such unique cases, I think, accelerated approval provides a pathway for drug access for patients.
It's about nuances. I think that's really important.
Exactly.
So,
exactly.
Look, it's been an hour. We've been chatting. This is a lot of fun. I think we talked a lot about the regulatory approval, accelerated pathways, synthetic control arms, a wide range of things. Anything else we should address on today's podcast? It's not going to be the last time you come on the podcast, but at least on that particular topic. Anything else maybe we should have talked about and I completely missed?
I think we hit most of the points here. I think we spoke about precision oncology, tissue agnostic approvals, and also, real world synthetic control arms. Again, this is a nascent field, right? I think this real world data is a synthetic, again, 5 years ago, I did not know what this was. And we all learned about this. This is implementing external control arms requires money, time, and resources. Again, evidence generation here is ripe for innovation in the digital era. And with the expanded use of real-world evidence for a clearer example. So again, we are still scratching the surface. So people say it's too imperfect to be used as a comparator arm to a clinical trial, which I agree. But that hurdle can be overcome. I think, with brilliant statisticians and brilliant designs, I think we can all overcome this.
Well, I'm a big believer. I'm a big believer in real-world evidence. To be honest, I think I believe. I actually was criticized before a lot about real-world evidence, and sometimes on social media when I talk about it, people will tweet back at me and say, well, are you saying we live in the unreal world? I remember exactly this happened to me several years back, and I wrote a paper addressing that. But you're right. I think it's going to be, it's here to stay. It's important. And I think we need to figure out how do we really try to answer questions as eloquently and as cleanly as possible, because there's a lot of noise and mess, and we have to make sure that we actually overcome that.
Of course, absolutely. This must be rigorously vetted by pre-specified detailed protocol, as we do for prospective clinical trials, right? It's an evolving area of methodological advancement and regulatory interest and statistical interest and clinical interest. So, again, many patients may not need to be treated with a non-therapeutic intent, non-therapeutic arms, especially with incorporation of real-world data. So that is what ultimately the goal of us in oncology is, to make sure that we have access, provide the right patients the right drugs, right? So again, these mechanisms are all exist to provide drugs to patients.
Well, Dr. Vivek Subbiah, the myth, the legend. Thank you so much for coming on Healthcare Unfiltered. Really appreciate it.
Thank you so much. I think we have a lot to talk to, but this is fantastic. Even I'm able to start thinking a lot just doing this podcast. Thank you so much.